Welcome back, everybody. So this video, the previous video, we've talked about how triglycerides are transported from the liver by VLDL into the adipose tissue. Now we're in the adipose tissue. Now imagine the body runs out of glucose or needs ATP, so it reaches out for its second energy source, which is fats. So what it does is actually tells itself to break down the fats that are stored in adipose tissue in order for it to take it and use it as an energy source, whether using glycerol to be converted into glucose in gluconeogenesis, or using fats and beta oxidation fatty acids and beta oxidation to produce ATP. Okay, so now what happens in the adipose tissue? The first thing that we have to know is that we need to know what signals from the body stimulates the function within the adipose tissue, or the process within the adipose tissue. Okay, so the first thing we have to know is that when there's low glucose due to fasting or whatever the reason is, the solution is fat metabolism. And so this leads to production of glucose and leads to ATP production. Glucose production is from glycerol heads and ATP production from fatty acid chains. So the first thing the body would do is tell its pancreas or the adrenal gland to secrete hormones that would stimulate the adipose tissue to start breaking down the triglyceride. Okay, so now the pancreas would secrete glucagon, and the adrenal gland would secrete epinephrine or norepinephrine under the stimulus of the sympathetic nervous system in case of a fight or flight situation. Okay, so these hormones right now would go and act onto a cell receptor where they bind into, and this receptor is a G protein gated receptor. So glucagon, epinephrine, norepinephrine binds to it. GTP is activated. The subunits of these ATP, the alpha ones, the third one would go onto the step number 2, activating adenyl cyclase. Activating this cyclase would cause it to convert ATP into CAMPs, and a lot of CAMPs would then activate PKAs. Now PKAs would then act on something called perilipin, by phosphorylating it, and then this would in turn activate something called CGI here in yellow. CGI then goes to act or activate an enzyme called adipose triglyceride lipase. And so adipose triglyceride lipase would now act on the stored triglyceride molecules. And so I've drawn here a ball-shaped molecule with fatty acid chains, 3 of them. So this adipose triglyceride lipase would act on this molecule to break the first fatty acid chain. Sorry for my voice, I've been screaming a lot in my soccer game. That's why it's sounding like this, but yeah. So adipose triglyceride lipase would act on cutting the first fatty acid chain and releasing it as a free fatty acid. Okay, the result is a free fatty acid and DAG, which is diacylglycerol. So it's a glycerol head with 2 fatty acid chains. Then the PKA would then activate an enzyme called hormone sensitive lipase. So any enzyme that cuts is actually a lipase. So the second one is called hormone sensitive lipase. This one would then act on DAG, cutting its second fatty acid to give us another free fatty acid plus a molecule called MAG. So MAG is monoacyl glyceride. So now we have 2 fatty acids free and 1 MAG. So now activation for a third enzyme that would cut and separate those 2 things, the glycerol head from the fatty acid tail, the third one. And so the product is a glycerol head and 3 fatty acid tails. And so just to keep in mind that the bonds broken within those 3 structure is an ester bond. Okay, so now when we have those products ready to be moved on, they will sure be moved on using lipoproteins, as you guys will learn in the future, that transport those things to either the fatty acids cannot leave the adipose tissue into the blood. They have to join with a protein called albumin, and so into the blood, and so albumin will then take them into either the liver or the muscle. So in the liver, they will be used to generate ATP, and also in the muscle will be used to generate ATP under the effect of beta oxidation. And then finally, the glycerol head would just move out on its own and then take into the liver under the effect of gluconeogenesis to produce glucose. And so we have successfully attained our objectives: production of ATP from fatty acid chains and production of glucose from glycerol by gluconeogenesis. So this is how the body deals with its low glucose. It would tell its adipose tissue to break down these triglycerides to release those glycerol molecules and fatty acid to either produce ATP by beta oxidation or glucose by gluconeogenesis.